Welcome back to the Point Blue Marine Lab. While we wait for people to trickle in, we're just going to let you know about our upcoming lives. We have 2 more. So our next one for next month will actually include a new grad student working with Point Blue, and they will be helping us with our California sea lion diet project. So that's next in February. So look out in the events page for that. And then in March will be our final live stream as our host.
So sad.
Because I will be leaving in the middle of March.
Yeah.
But that one will be really fun, really interactive. We will be talking about 2020 data results, maybe a little trivia game, and there might be some prizes included, so look out for that.
Yeah.
And just a Q&A with us in case you guys are curious about what we're going to do after this and whatnot. So make sure to check out Point Blue's events page for upcoming lives.
Looks like Meredith's here. Hi, Meredith.
Hello,
And
Michelle. She's sad that our lives are ending.
I know, me
But
too.
They've been really
So
fun.
a few more, so yeah, stay tuned. All right, we'll let some people trickle in.
Yeah, thanks for showing up.
Yeah, thanks for coming.
This one's really exciting. No poop or puke this time,
No.
so you can eat your lunch in peace.
Yeah.
Quinn is
So,
here.
Let's go.
Yes. Our loyal viewers.
Mm-hmm. Oh,
Meredith, I'll be sad,
sad.
too.
yeah.
It's been really
I miss
fun.
the lab. It's like Holly's here. Hi.
From Gilbert, Arizona.
Wow,
Whoa,
cool. Thanks for tuning in. Great.
Sarah.
So this is hi.
Mm-hmm.
Hi, everyone.
Welcome, welcome.
Wow.
Well, we'll give them a little more time.
One more time.
But basically, we'll be talking about some
Zooplankton.
zooplankton. Look at those guys.
Yeah. Fun times. All right. Guess we should start.
Yeah.
All right. Hi, everyone. I'm Olivia.
Rebecca.
Welcome back to the Point Blue Marine Lab. So, as you know, I started here back in September of 2019 after graduating from California State University, Monterey Bay, where I was a NOAA research fellow. So, actually, a part of that program, I got to go on a research cruise, and that is where I met Point Blue biologists. And so, this is kind of fun because this is kind of how I started with Point Blue on one of these zooplankton research cruises. And since my time here, I have decided I want to pursue a master's degree in fisheries science at Oregon State University, where I'll be looking at microplastics in myctopods.
Yeah, and I started here in January of 2020, and I'll be here until the middle of March. I graduated from Humboldt State University May of 2019 with a wildlife biology degree, and I've since had various positions working with marine wildlife, specifically seabirds. And so once this position is finished, I hope to also attend graduate school at Oregon State University, and there I will be studying the Brandt's cormorant population on the Farallon Islands.
Yeah, and unfortunately, due to technical difficulties, our lab manager, Meredith Elliott, will be in the comments answering questions, but she won't be here over video today. So keep an eye out for her responses if we don't get to them right away. Yeah, so Rebecca and I, as you know, we live together, and we've been following all the COVID guidelines, so that's why we can work together without masks here in the lab, which remains closed to the public, unfortunately, but we look forward to when it can reopen. And yeah, for those of you who missed our last live streams, those should still be up on Point Blue's Facebook page, so go check those out. Yeah, so as you may sure. Hi everyone.
Sorry about that.
Can you see us? We're back. Sorry.
It's tradition.
Yeah.
For technical difficulties.
Can you see
Let
us?
us know.
Yeah. Okay, so I hope you guys heard my spiel about the NOAA cruises, or the research cruises.
Okay.
This is one of the ships that I actually went on during my time on one of the cruises. But yeah, so these are reoccurring surveys that are along the California coast, and they have established transects in Cordell Bank, Monterey Bay, and Greater Farallones National Marine Sanctuaries. And so these provide a long history of data collected at these points, so we can use these to kind of predict future oceanic conditions and distribution of wildlife. And so some of the management issues that these cruises focus on are preventing whale ship strikes and entanglements, monitoring wildlife hotspots, and also monitoring ocean acidification. So along with collecting the water properties on these cruises, we also collect the biological specimens, which is what we're talking about today. So actually, on these ships there are these big nets called a tucker trawl, and then these are at the end of each of those nets. And so the ship will use acoustic data to target swarms of krill specifically. So krill are an important ecological indicator species, and they can be found using this acoustic data. And so we will be using this net, which actually is composed of 3 separate nets that can open and close at different depths. When we send the messenger, there's this little contraption that goes down the line and can open and close these nets here. So we can get a different depth profile in different species that are found in one single toe. So that's really cool. But yeah, so all those critters will be collected into the end of the net. This is called a cod end. And once it's on the boat, we'll rinse down all the nets and get all of the biological samples in here. And then we'll take this and we'll transfer all these guys into a jar like this. And then you can see there's some krill in there. And so these are preserved in formalin, so we can bring them back to the lab to analyze. Yeah, as you can see, there are lots of critters floating around in here, not just krill. But I'm going to turn it over to Rebecca to talk more about krill specifically.
Yeah, so I'm sure most of you have heard of krill in your lifetime, maybe from a science class or from our live streams or even from Finding Nemo. But the word krill actually comes from the Norwegian word krill, which means a small fish or a small fry, which is quite fitting. And so, yes, this is what they look like. And there are about 85 known species of krill worldwide, and they all measure average 2.5 inches, which is like a human pinky. And so they are shrimp-like in appearance, as you can tell, but we can distinguish them from shrimp by the presence of external gills under their thorax, and I think they're more cute, in my opinion. So yeah, let's talk about some anatomy of krill. This is your average krill. They have this head part called the cephalothorax, and then the abdomen, which is these segmented parts that contain the swimming legs. And so this is what they look like, and then under a microscope we look for certain characteristics that can help us key it out to species. And so I'm just going to go through a couple that we might find in today's sample and how we can tell them apart from each other, because at first glance they look the same. And so there are certain things we look out for when we have them under the microscope. One being the rostrum. So that is this pointy part, sometimes pointy, this little nose above the eyes. So in Thysannoessa spinifera, also known as T-spin, it's got this really strong pointy rostrum. And then with Nematocellus difficilis, also known as NDiff, it's really not a strong rostrum, it's kind of flappy. And then in Euphausia pacifica, also known as EPAC, it's not really there. So that can really tell us what species we're looking at. Another thing is the 6th segment on the abdomen. In EPAC and NDiff, it's way longer than the other 5, whereas on T-spin, it's kind of the same all throughout the body, and they have these really pokey spines running across the back. T-spin's a pokey guy. And so those are just a few things we look for when we're identifying to species. And then we also can sex adults. It's way harder to sex any juveniles or immatures. So when they're an adult, we look for a few things. Male EPAC have this fleshy part on their first swimmer leg called the patasma. And then sometimes males will have this little sac on the feeding legs, and that is the spermatophore. You guessed it, it contains sperm. And so sometimes it'll be already transferred to the female, so females may have this, but it's just something we look for when we're sexing. And then if those are not there, and so we flip this guy on its back, and then we look for this little flappy part called the thalicum, and that can tell us that it's a female. And so we also can identify what age class each individual falls under by measuring using this little guy or the ruler in the microscope. It can tell us how long it is, and then based on that measurement we can tell whether it's an adult, a juvenile, or an immature. And so yeah, that's what we do when we pick out our little krill friends. And then also they have these really cool organs called photophores, which actually emit light. They look like little spaceships. And the exact function of this organ is unknown, but it could be for mating purposes, social interaction, or maybe even camouflage from the ambient light above. So yeah, I think that's a really cool function of these little guys. And so they occur worldwide, which is great because, like Olivia mentioned, they're an essential part of the marine food web. Everything from fish, seabirds, and the biggest animal in the world, the blue whale, consume these in large quantities. So it's great that they're just available everywhere. And so what do they eat? They are filter feeders. They use their feeding legs to sieve through the ocean looking for phytoplankton usually, which is tiny marine algae. And actually by eating and excreting carbon and nutrient-rich pellets, their poop, that sink to the sea floor, they are actually an important influence on the carbon cycle. So not only are they a great food source for all marine wildlife, they actually assist us in global climate change. So we love these little guys. And so they're mostly found in these swarms. These are all krill. Reaching up to 30,000 individuals per swarm, and they undergo a daily migration, meaning during the night or during the daytime they spend their time really deep in the ocean, and then at night they migrate up the water column to feed near the surface. And so marine predators take advantage of this daily migration, such as the Cassin's auklet that we talked about last live stream, feeding on these krill when they are so close to the ocean surface. And humans take advantage of this daily migration, as Olivia mentioned, to scoop up a large quantity of these populations in order to further analyze them in labs, which we do here. And so why do we study zooplankton communities? By studying the abundance, the species, and the age structure of these zooplankton communities, we can understand ocean productivity and how properties in the ocean are changing in any given year. For example, from 2005 to 2006, krill abundance was really low. And during those years, Cassin's auklet, that seabird we talked about last live stream, failed to breed successfully. And so getting a detailed look at the available prey and connecting that to breeding productivity to certain marine predators, we can understand what ocean conditions are both beneficial and detrimental to marine wildlife. And this ultimately assists biologists in preparing and predicting how the future will affect certain species in order to successfully conserve them. And so krill are not the only thing that get stuck in that net when we do our Tucker trawls. There are also a ton of other tiny creatures we find. So now Olivia's going to talk about those.
Yeah. So as you can see, when it comes out of the net, it's quite a soup of different organisms. So you can see there's some fish there. All these clear spots are like jellies. Yeah, so we gotta remove the krill from this soup. But some of the other organisms we're likely to find are copepods. So these are going to be these small pill-shaped creatures. These are really abundant in the samples. We don't have scopes powerful enough to identify these species, but sometimes we'll send them off to be identified. But yeah, so we'll be removing the krill from kind of this group of other things. Some other things we'll find are these fish larvae, so they're going to be really tiny. Also the larval stages of cephalopods, so like octopuses. And then also these two groups, they're decapods, so these are the shrimp-like decapods and the crab-like decapods. And so these actually, we use this big binder right here to key these out to species because they look very different from their adult forms. So you guys might be familiar with this one here. That is when you're at the beach and if you dig near the waves, you might find these like sand crabs. That's actually a baby one of those. Yeah, but they have like these large spines that come out. They look really funny. But yeah, and then yeah, with the fish larvae, we can't really identify those species unless they're big enough. But one family of fish we do identify are Decapods. So these are those lanternfish. You can see they have the photophores like the krill. They also do that vertical migration at night to feed on the krill. And these are actually going to be my study species in graduate school, where I'll be looking for bioaccumulation of microplastics in them. So yeah, they're very cool. They got like these huge eyes, these photophores, very cool. And then one last thing we're probably going to find are these. So these are called Limacina. They're actually going to be much smaller than this. They're little tiny shells that we'll see in there. They use these flaps to kind of fly around in the currents. But these are important indicators of ocean acidification. They have a calcium carbonate shell which can dissolve if the ocean is too acidic. So making note of these in our sample is really important to do those long-term, tracking those changes over time. So yeah, with that, we will get right into showing you how we do each of these steps in this process.
Yeah. Feel free to ask questions. Remember, look out for Meredith Elliott in the comments. She's our senior scientist here in the lab. She knows a lot about our projects. Yeah. All right, here's the kickback.
So Rebecca is going to be showing you what we do in the fume hood.
Yeah, so like Olivia mentioned, these samples are preserved in formalin, which is a really toxic chemical. So we like to do this in the fume hood because it is carcinogenic. So here is that jar that comes straight off that cruise and comes into here in the lab. So we got our sieve that we're gonna pour it into. We tare this so we get a measurement of the sieve itself. And now it's ready to be full of krill and other things. So we put it in this funnel and a jar labeled formalin so we don't cross-contaminate anything. And we pour it in carefully. This is just gonna filter out the formalin and capture all of our krill. So we wanna make sure it doesn't overflow, because that would be really bad. See all those little guys? So we just kind of drain it off. Let's see if we can get a little more. Okay, so now there's not a lot of formalin left, so we're gonna pour it back into the jar to get all those remaining krill.
Hope y'all can see that.
So we're gonna pour it back in the jar. Put the funnel back on. Put the sieve back in. And now we pour again. Let it drain. We can also use some forceps to break up the water tension. We got lots of krill. You see, it's kind of hard in the fume hood. But yeah, look at that soup. Yeah. So we have a sample prepped, so I'm just going to continue. There's still some in there, but we'll move on to the next step. So then we weigh this, get a wet weight of the krill with our scale, and then we would record that on the data sheet. And then we move it over to our next funnel here, where we will rinse it and try to get as much formalin out of there as possible. So rinse it into this beaker and it'll drain off. And then also in here, let's see if I can get it, but of course there's this label telling us what year, what net was used, a bunch of information about this collection. And then if that happens to get destroyed, or if it's hard to read, there's actually another paper in here, if I can get it. Hold on. Oh man. Yeah. So here, can show us all that's on this outside label. So it's important that we get that for our data sheet. So yeah, while this drains, we rinse and rinse until the water kind of runs clear. We can use our forceps to break it up. Okay, so from there, we bring this guy over. And we want to get the volume. So we will dump it in a beaker. Just plop. And then we will fill it up with water until the krill are just like at the bottom of the water in the beaker. It's a long process, but worth it.
Yeah, you can see all this krill and those other things floating around in there.
So then we let them settle to the bottom. And then we'll kind of measure where it ends, the top. So,
I'd say it's about 300 milliliters.
yeah. So that is how we take the volume. And then, an important part before we split, we kind of pour it into another tray. And we look for big organisms, such as big fish, some big shrimpies, because we don't want them to get caught in the splitter. It can
skew the data.
Yeah.
If there's large things in here.
But it looks like all it looks just all the right size we want. So now we already have a sample placed in the splitter. So now we want to split these big samples down to about 100 krill.
Yeah.
And to do that,
So, yeah, as you can see, so I've already split this a few times. As you can see, there's a large quantity of krill, and it would be nearly impossible for us to identify, weigh, measure all of these krill. So we do this splitting process to get it down to about 100 krill, like Rebecca said. So that's like a reasonable amount of krill that we can kind of extrapolate and guess what the rest of the sample was probably mostly comprised of. So yeah, I've already done, up here we have our little Hulk, and with this guy we count the number of times we split this in half, and we move it each time, and then we flip them so we know which side to take. Yeah, so this is our splitter. We're already at split 4, so this will be split number 5. Yeah, if you can see that. So there's a bunch of the krill left in here that came from this sample. So we kind of rock it back and forth, mix it up. And yeah, so I'll be dumping it into these two bins down here, you can see. Yeah, and down the middle of this, there's kind of a piece of plastic, you can see there, that will split this in half, or roughly half. So, dump it out. Then we just rinse it, try to get things from both sides down the middle. Yeah, so now we have a left and a right split. So this comes out and there's two separate containers. You can see that. So this is approximately half of what was in here. And according to the Hulk, since he's facing forward, we will be keeping the left side, and then we'll split this left side again. So I'll take that side, and then all this, you can see there's probably more than 100 krill in there. It's kind of hard to tell. But after doing a bunch of these, you can tell like what's about 100 krill. So I will be pouring these to the rest of the sample. So yeah, now this will get dumped back in here. I'll split it again. To flip this guy. So we've done 5 splits now. So this is number 6. And then this, we just want to add enough water so it can get mixed around and split evenly.
Yeah, and then the rest of this krill just gets bottled up again. No krill wasted.
Give this another split. Alright. So, if you could see in here, looks like about 100 krill to me. So now we can take, so since we kept the left side last time, we're going to take the right side to look at. So
yeah, we'll take it over to the microscope.
Yeah.
I think Michelle had a question about why her internet keeps freezing. Why do you split it multiple times? The sample is like really big. This is just part of it. And so we split it in order to get about 100 krill. And that is just a good amount for us to do that identifying the species, sexing the krill, and all that. Yeah, the goal is to get a representative of the population, not the whole shebang. And so we'll take you over here to the microscope, flip you around, if it'll flip, set you up. And so while Olivia gets some of that krill in a dish, I'll just show you a few things we picked out when we were prepping that sample. Zoom out a little. So these are some fish that we found when we were searching for bigger individuals. So yeah, we'll pull those out. And then we actually got a shrimp. Look at those eyes. Oh. Oh, maybe push this up. There we go.
That's better.
Yeah, super cool eyes. And then I'll pull out a krill to show them side by side, so you can really see those differences.
They do look the same. But yeah, take a look at those eyes. They're a lot
very
bigger on the
different.
krill.
And then, yeah, check it out. Really cool. And then just like the bottom part is a little different. But yeah, those eyes really tell us. I mean, there's some krill that have that kind of eye, but this has more different characteristics. So yeah, those are just a few things we pulled out initially. And then here's...
This is, yeah, what the sample looks like under the microscope. So this just came from that splitter. And as you can see, we got quite a few krill here. But then also, we can take a look at these. These are those copepods I was talking about. Those little pill-shaped animals. So yeah, they're really small in comparison, but there's going to be a lot of those in the sample. Yeah. So they're actually very nutrient rich. They're filled with lots of oils and stuff, which is good for predators. But yeah, we'll focus on the krill for now. And let's see if there's anything else in here. Looks like mostly krill.
Yeah, so we would pull out all those krill
Mm-hmm.
and then measure them, identify them, and whatnot.
Yeah. So this actually here, this long stringy thing, it's called an arrowworm. And they actually eat krill, like the baby krill. So we got a krill predator in here, which is fun. And then I think I saw, is that one? Let me try to zoom in there. Oh, actually, it's not what I thought it was. Trying to find one of those Limacina, but...
Debbie's asking what magnification we are using.
This currently is 10 times what we would see with our eyes.
Yeah.
Yeah, Ketognaths, that's also what the arrowworm is called, a ketognath.
Yeah, they got crazy mouth.
Mm-hmm. Yeah, and if you can see in here, so this is like an adult krill. There's also these zoea. So these are younger krill that have yet to fully form. Yeah, so those are just some other things in this sample. Oh, right here in the middle, this is actually a protozoea. So it's like before the eyes come out of the carapace there. This is actually an even younger krill than the zoea. And these are important to note because it means that these krill are spawning. So yeah, there's little baby krill in here. There's another one. Yeah, oh, right there. This is what I was looking for. Is this shell? I think that's one. Yeah. So there are these are those Limacina, those snails I was talking about. These are important to note because of ocean acidification. So you can kind of see the spiral there. Yeah, so on our data sheet, we just have a box we check that there is Limacina present.
Mm-hmm. Yeah,
So,
good
yeah.
variety of krill.
Mm-hmm.
Some big ones.
Yeah, so these are adult krill here.
Do you know what species with that?
I think this is EPAC. Let's take a closer look. Yeah, so first thing I look for is the rostrum. And to me, that looks like a very short rostrum that doesn't extend past the eyes. You can see it kind of ends there. Yeah, so another thing, maybe we can
See the denticle.
see the denticle. So I think Rebecca mentioned they have this hook-like thing on their carapace. I wonder if I can get him. So I'll flip it over. It's a lot harder to do through the camera.
I also have the needle.
Yeah. Okay, got it. Oh. Well.
Sometimes it's not there.
Sometimes it's hard to see. Yeah, we kind of pry this part back to look for a denticle. These are also fairly old krill. I think it might be a little too hard to see on here, but yeah, there's
our last live stream we showed the denticle. We can show the longer sixth segment.
Yeah, so this one lacks those spines that we would find on T-spin, but it has this long sixth segment here. Yeah, big male E-pack. I agree. So yeah, the next thing we look for is the sex. So now that we know that this is E-pack, we'll look for the sex. So this is that petasma, that fleshy bit on the first swimming legs that we were talking about. I guess to the untrained eye, it doesn't look that fleshy, but on the females, they're just like little sticks. This guy has kind of some extra little flesh parts right there. And then also its gonads. You can kind of see it's that
little
orange tint. Let's see if I can flip them over. Oh yeah. Okay. So yeah, you can see those are the gonads there.
Yeah.
You can kind of see it. And then a female would have like a little flap right in here that the male would deposit that spermatophore to fertilize it. But it's not here because it's a male. Yeah. So once we've identified the species and the sex, we will go ahead and measure them. Should we bring it
Maybe
off?
we should show the decapods.
Oh yeah, one more thing before we move out of the microscope. We'll do those other things I was talking about, those decapods that we use that book to ID. Here are some examples.
They're so cute.
These are baby crabs. They're megalope phase. So the zoea stage would be even smaller than these and less developed. But yeah, these are
Baby
crabs that are floating around with the krill swarms.
Yeah, they got caught. Poor things.
Yeah, I think there's some other... Oh yeah, so this funny looking one here has an even smaller crab. So it doesn't even quite look like a crab. Wow, it's not wanting to focus. Yeah.
Yeah, look at that tail.
Yeah, so these are decapods. Look at him.
Yeah, really cute.
Yeah, so can you believe this turns into a crab?
Nope.
So funny. I think, here's a shrimp-like decapod. Again, these kind of look like krill, but they're different. There, you can see him.
Yeah.
So yeah, that's kind of what we'll find in these samples. And now we'll show you how we measure them.
Okay. So, with bigger guys, we use this fancy equipment. With the smaller ones, we can use the microscope ruler. But, so we'll pull out each krill that we find. And we'll put it on a little dish. Try to get it as flat as we can.
I think it's so cute how we set them up like that. They're very...
Yeah.
Look at them. So cute.
So yeah, then we take this and we zero it out. And we start at the eyes and we move it all the way to the end of the tail. So yeah, it's just like an example. And then this will tell us what size it is. And then we'll use that table and it can, when it falls under a certain category, we can know if it's an adult, juvenile, what age class it falls under.
Yeah, and then this one was in the 21 point something range. So we put them, all the ones that are like 21 millimeters will be put into each of these squares, and then we'll weigh those together and then divide by that count to get the average weight of each of these size classes.
Yeah, so all these squares will have like
a bunch, like a stack of krill. Yeah, so we'll go through this whole sample, identify species, and then we'll put them in these little dishes.
Yeah, we'll weigh them, write it on our data sheet.
Yeah.
Yeah.
So, keep this going up. Yeah, now, sorry we kind of neglected you. We didn't see the questions. But
I think Meredith is holding it down.
Awesome. How do we switch it?
So yeah, let us know if you guys have any questions about the process, about krill, about the cruises. This sample that we just showed you is from 2018. I'm so red.
I know.
It's hot in here.
Pink. Okay.
Our heater's on.
So let's see. Oh, way back when Quinn said, "Why do krill swarm like that? Does it attract predators?"
Oh, the swarms.
Oh yeah, Meredith answered that briefly too. They need to find each other to reproduce. And that's a big reason why they do it. Yes. Also makes them it makes them susceptible to predation. Yeah.
Yeah, it's kind of sad, but they want to find that phytoplankton. So, and then, I mean, yeah, it's kind of bad that they're all together, but
keeps them from being singled out as prey, I think.
Safety in numbers. So, like, if one krill saw a predator, maybe they could all, like, swim away instead of not paying attention or something.
Yeah. I was also watching a video recently on why, like, they think fish swarm like that, too. And it actually uses less energy for them to swim because they're with a whole group, so they don't have to expend as much energy. Which, I don't know if that's true for krill, but just a thought. Yeah.
Yeah, we answered that one.
Oh yeah, flatfish. Yeah, those were fish. The fish we saw were flatfish. What magnification? Okay.
Yeah. Glad you guys think these are all
cute.
We
love them.
What's the longest one?
Oh wow. This goes up to 24, but I want to say I found like one that was 26 millimeters long.
Yeah, so that would be like that.
Yeah. It's kind of hard to tell.
Let's see. Almost the average length.
Yeah.
Remember, there's 85 species, so the average length is like a human pinky swimming around.
Yeah, the ones that the penguins eat in Antarctica, they're like huge krill. They got thick carapaces.
Yeah, so they range, but we usually just get
the EPAC, the smaller
up to
ones.
24 millimeters.
Yeah, and sometimes they're really tiny. The zoea generally range from like 2.5 millimeters to 5, and that's really hard to tell like what species it is because certain features haven't fully developed. But yeah, they could be that small, and we can identify them to species.
Yeah, it's a challenge when they're not adults.
Meredith says maybe somewhere in the 27 millimeter range. Yeah, I'd believe it. Yeah, they can get big.
Chonk.
Especially T-spin, the one with the spines on the back, can be like really thick, big boys. Yeah.
Krill are abundant but patchy in the ocean, so it's not super easy for predators to find them all the time. Yeah. Gotta be there at the right time. Those whales.
Yeah, it looks like we're kind of over the time limit, but if there's any last questions, feel free to ask us.
Yeah, and fun fact, krill can live up to 10 years. I think the average is about 6, but 10 years.
I had no idea.
And even 6 being the average age 6, that's a long time for being such a sought after prey from so many predators.
Yeah. And didn't you say one female can lay 10,000 eggs?
Yeah. And then they can spawn more times during the spawn season. So they're just pumping out the krill. They need to feed the ocean.
Super cool. But yeah, they're very important species.
Yeah, we love them.
Yeah. Krilling it. All right, it looks like there's no questions. Thanks all for tuning in.
Yeah, tune in next time to see our sea lion diet study. And Ashley said 10 years. Yeah, it's a long time.
Yeah.
It's like a dog. So yeah, look out for our upcoming lives. Feel free to share this with your friends. And just...
Yeah. And of course the donation link is on this video, so if you like what you're watching, feel free to donate for ocean conservation, and we really appreciate you all watching.
Yeah.
We'll see you next month.
Krilling it.